so now we want to take a look at translation. So we just finished up the transcription video. We now know how to go from DNA to RNA. Now we want to be able to go from RNA to a protein, and those proteins will ultimately make up traits. So from translation, we're going from a nucleic acid language to an amino acid language. So in here, how does an mRNA code for proteins? And you should have seen this in your regular bio class, but just in case you forget, say this was our sequence of DNA. The T would bind up to an A, the A would bind up to a U, the C would bind up to a G. We make our mRNA sequence here. And then, and we'll get a little bit ahead of ourselves, but every 3 letters, which is going to be called a codon, will code for a very specific amino acid. So every 3 letters codes for these amino acids. These are going to get hooked together, and they'll make a very specific protein. Okay, so I know we got a little ahead of ourselves with a couple of the terms, but we'll take a look at this. And again, that shows you the codons and how that works. So cracking the code, Crick determined that this idea of a codon system, you can even see it on the screen, that if I were to give you all these letters, those letters would just say nothing until I coded for every 3. Then it makes sense. Why did the red bat eat the fat rat? That is a sentence that, while it's goofy, actually makes sense. It can work. But if I didn't highlight any of those letters, you'd be like, well, what am I looking at? What's this say? So every 3 letters is going to be kind of like a word in the sentence. Nirenberg and Khorana, 2 scientists, also determined that there's this mRNA amino acid match, and that for every mRNA sequence, every codon, there's an amino acid that matched that. So they added these fabricated mRNAs to a test tube of ribosomes, tRNA and amino acids, and they were able to actually create artificial mRNA. They found that UUU coded for phenylalanine. So through their research, we know that there's this codon chart that we can go look at and say, okay, what codon do I have? What codon does that match up to? So this is your code. This is the table. You're probably slightly familiar with it, and it's the code for all life. It's very redundant, and that's an important piece to know. This idea of being redundant is that we have several different codons that code for each amino acid. So if you look for, like, say, leucine, leucine could be CUU, CUC, CUA, CUG, UUG, UUA. There's a lot of versions to get leucine. Why is that important? Well, let's say your code was CUU, and that's how you made leucine. If you were to change that to CUC, you had a mutation in your DNA. You notice that it would still make leucine? So you might be sitting there right now listening to this with a mutation in your sequence, and you would never know, because different sequences still code for the same amino acid, still makes that same protein. So it's important in this redundancy of code that not every last thing is only one way. You can code, say, proline in several different ways, and that's important. That third base, so it's like CUU, CUC, CUA, CUG, that third base, because it can be a bit more variable and still code for the same amino acid, is called the wobble sequence. A fancy, funny name that they gave it, but you probably will see that word come up in your readings. The wobble sequence is what causes it, so you can create the same amino acid from different codes. It's also very important to show that there is only one start codon. As we try to make proteins and we start hooking these amino acids, you can't make a protein unless you start with the methionine, or the start codon, which is AUG. You do need to know that, because you could look through a billion letters, and until you come across AUG, that protein can't start to be made. You will keep going, coding every amino acid that you come by until you reach either a UAA, a UAG, or a UGA. Those 3 are your stop codons, and those stop codons will say, no more, you're done. And we'll find out later when we talk about mutations that some mutations in your DNA will code for a premature stop, and that could be very dangerous because you haven't made the entire protein yet. But these are very important to know, those 3. You don't have to memorize them. You will always have access to this table. However, knowing them makes it a little simpler as you're looking at it, just to know those. But make sure you know your starts, make sure you know your stops. So how are the codons matched to the amino acids? So if this is my DNA, again, here's my 3' to 5' template. I'm going to replicate 5' to 3', so here's all my letters that I just coded for with mRNA. Each of these correspond as a codon to a very specific tRNA, which is your anticodon. So AUG The anticodon is UAC. What's important about these anticodons is that they are carrying your amino acids. So they're little molecules floating around in the cytoplasm that are carrying, say UAC would be carrying the methionine amino acid. So as soon as it finds an AUG, AUG hooks up with UAC and it will release this methionine apart. It will release it off the tRNA and I can start to make my protein sequences. So it's very important that for every codon, you have an anticodon. Those anticodons are carrying the amino acids, and every time these start to hook together, the methionine will connect to your arginine, which will connect to your valine, and this, now we're actually starting to create a protein. Okay. So again, we're working through this. This is the picture, it's called a cloverleaf, but specifically in the tRNA, we have this anticodon that's hooking up to the amino acids. You'll notice the anticodon is down here at the bottom. And then I always remember this is the tRNA has the anticodons because it looks like a T. There's your T right there. And then right up here, this is your amino acid attachment site. Every anticodon is carrying one of those 20 amino acids, and they're looking for their codon complement so they know what to release. And again, you can see just different structures of the anticodon and how they work. One important enzyme is this enzyme called aminoacyl tRNA synthetase. This is the enzyme that bonds the amino acids to tRNA. So it does require a little bit of energy, but say we want to put methionine on that, that anticodon, this enzyme is going to be what hooks it together. It's going to release, as soon as the codon and the anticodon come together, they're going to release, and this enzyme is going to hook another amino acid back onto that tRNA. So it's important so that it's able to come on, and it's also able to release the amino acid at the ribosome easily. So you might see that word in your readings or in some of the test questions. Now we're at the ribosome, so you get the idea of tRNA meeting up with mRNA. Now we want to actually look specifically at the ribosome to see how this all works. So in the ribosome, we're going to facilitate the coupling of the anticodon and the codon together. If I look at the specific structure of a ribosome, I see there's actually two parts. There's the large subunit and the small subunit. And those two together, whenever you're ready to hook the codon and anticodon together, these two parts come together and they start to decode and make those amino acids. It's very important, I'm going to come back up to this, that you see that there's actually 3 sites in the ribosome. There's an A site, there's a P site, and there's an E site. In the A site, this is the aminoacyl tRNA synthetase site. This is what's going to be holding the tRNA. It's kind of like on deck in baseball terms. It's holding the tRNA that's carrying the next amino acid to be added to the chain. The P site, the peptidyl tRNA site, is going to be where we actually hook the codon and anticodon together. So it's holding the tRNA to the growing polypeptide chain, and the E site is where it's going to empty it out. It's going to leave, the tRNA is going to leave the ribosome from the exit site. It no longer has the amino acid connected to it, but it's going to leave and it's going to go get hooked onto another amino acid back in the cytoplasm. So it's very important to understand that the tRNA comes in at the A site, it bonds at the P site, and right at this P site, it's going to release the amino acid into the chain, and then this empty tRNA is going to move over to the E, and it's going to exit. So in terms of protein targeting, again, this is showing you how they can hook together, how they can release different parts to the cell. But this, you should be able to talk about this. Before we get to this, I do want to pull up an actual animation. I'm going to put this on Schoology, but I want to be able to show it to you in here as well. I want to show this to you because it's a really good one. That let's actually take this out. I want to show you this because I want to be able to walk you through it. So this is how translation works. I'm going to stop and pause it as it goes through.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome.
Okay, so this is where you're seeing the large subunit and the small subunit are coming together. The mRNA is coming through, and as you can see, we're going to code the first piece at the P site. The A site's the one that's on deck.
The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time.
So you see right there, as this is moving to the E site, you saw that the methionine is now hooked up, so that when this moves into the P site, we now have 2 amino acids hooked together. You're going to see another one come in at the A site as they're on deck.
As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
And so from there you can see that now we had the start one that started it. The stop one doesn't actually code to a tRNA. So in that we're done, this is the protein, this is what we're trying to create. So as we come back up to here, would you be able to, and even if you want a snapshot, I'm going to put it up as a thing here. If you just took a snapshot of that, could you explain, this is always your self-check, could you explain going from DNA to how we synthesize mRNA, to how we do our post-transcriptional processing, how it wiggles out and goes to the cytoplasm, how the tRNA here is hooking up, the mRNA is coming through, it's meeting the ribosome, we're going through that whole process, and we're making a protein. If not, I would pause it right there and see if you can actually tell the story. And in about 2 seconds, I'm going to show you how to do it. Okay, so you have already paused it, so hopefully you got a chance to go through it. So again, we take a look here, RNA polymerase is the part that is actually going to start transcription. Remember, that's the one that's going to go try to find the TATA box in the promoter, and it's going to start that transcription down the line. It makes a pre-mRNA. Remember, pre-mRNA has introns and exons. The introns are the parts that we have to cut out. The exons are what we want to keep. That's the spliceosome made up of the SNRPs. And that's how we make a mature mRNA. So there's your exons, there's your introns, we cut them out. And to get them ready to go, we put a 5' GTP cap, we put a poly A tail. Now we have a mature mRNA. It is coming out into the cytoplasm. It's meeting up with the large and small subunit. And now we've made a ribosome. The mRNA is coming through. The tRNA is coming down and trying to find its match. The tRNA has the anticodon. The mRNA has the codon. When they come together, they come in at the A site, which is the on deck. They bond at the P site. They release their amino acid. Now the tRNA without the amino acid moves over to the E site and exits, and we keep going through this. You know, to start this, we have to have a methionine, and to end the protein sequence, we have to have one of the 3 stop codons. Once that's done, so there's our 5' and 3' side, our tRNA is matching up. Once we have all that working together, we create this polypeptide, it's released, and we've made a protein. So looking here at the transcriptional units, again, you can see the genes. There's your promoter with the TATA box. The enhancer is about 1,000 bases upstream. But again, just taking a look at how do we go through and do this, the TATA box is recognized by RNA polymerase. That then goes through, we start transcription there, we stop transcription there. We start coding down and creating an mRNA strand. We code out all of our, or we excise all of our introns. So there's our translational start, our translational stop. You have your UTR sections, which are your untranscribed regions. We have our pre-mRNA. We take out all of those introns. We have a mature mRNA. We put our GTP cap and our poly A tail. It's ready to go. Taking a second to look back now, you have to think this all happened because we started in the nucleus and we moved out to the ribosome. But what happens in prokaryotes when they don't have a nucleus? Where do we go there? We have DNA that's floating in the cytoplasm, but we don't have a nucleus. So we can see here we have bacterial chromosomes. That is floating around in the cytoplasm. We still have our cell wall and our cell membrane, but how do we get through there? We're going to go through, in our biotech unit, in the next unit, we're going to talk very specific about how bacteria regulate this. But just as a quick rundown of how they differ and how they both go through and do this, with prokaryotes we have DNA in the cytoplasm, in eukaryotes it's in the nucleus. We've said that. In prokaryotes it's actually a circular chromosome, where in eukaryotes it's a linear chromosome, it's double-stranded. In prokaryotes we call it naked DNA, whereas in eukaryotes it's wound onto the histone proteins. Histone proteins are simply little proteins that the DNA winds around to get tighter and tighter and tighter. And that way we can fit a lot of DNA in a very small space. Again, in prokaryotes it's naked. It's a circular chromosome. It's not wound around any specific proteins. Interestingly enough, in prokaryotes there's no introns. There's nothing there. It's simply they only have the exons. They only do what's expressed. Whereas in eukaryotes we have both. So it's kind of interesting, prokaryotes being as simplistic and as what we would call less evolved don't actually have any introns. They only have the expressed genes. Okay. So again, we're going to code those out in eukaryotes. As we go through translation in prokaryotes, they're pretty much simultaneous. The DNA is in the cytoplasm. There's no editing because we don't have to take out any introns. So the ribosome basically reads the mRNA as it's being transcribed. There's no, we're going to go through transcription and make it RNA and then transfer it down to do translation. As DNA is making mRNA, the mRNA is being made into proteins. So it's a bit more simultaneous in nature. So the difference again between the two is really just your time in physical separation. It takes eukaryotes about an hour to go from DNA to proteins with no RNA processing. So prokaryotes are a little quicker in that aspect. Okay? And that is translation. And again, your self-check should be, can you go back through and explain this to yourself, explain it to a friend? Can you go back through and talk about not only how we make a mature mRNA, but how then that makes a protein? Can you use the table or the codon wheel to come up with each codon for each of the sequences? But that's really translation in the basics. And of course, you'll get a chance to see this in your models as well tomorrow.